Thanks for that great welcoming and pleasure to be here with everyone. So mahalo for the invitation, and I'm going to focus mostly on my previous work, but Matt mentioned maybe I should give a little taste of what we're doing at CTAR and some of the vision for CTAR, so I'll plug that in at the end. But I wanted to, I think aligned with some of the work of this, why is this? Ooh. Technical difficulties. We tested it and it was working perfectly before. Now we're frozen. I think, yeah, even I can use the keyboard, but the keyboard's not advancing either.
Oh, keyboard's on?
Yeah.
Oh.
Sorry, guys. Oh, wait, there we go. Super delayed. Okay.
I think this is our.
Okay. Let's get to it. Sorry about that. So I am from Oahu, from Hawaii, born and raised. I did work at University of Hawaii back in the late 90s, before some of you were born, and doing endangered Hawaiian tree snail kahuli conservation with Mike Hadfield and the then zoology department. And my role as a research associate doing that work in the 90s was to coordinate our field work. So we did field surveys for these endangered species. We did predator exclusion. This is the first predator exclusion fence that was built in the Waianae Mountains. And then we did captive breeding here on campus. And we also did molecular genetics to figure out what are the populations, the evolutionary significant units that we really want to protect. So that was my first foray into conservation genetics. It got me interested in the field. I realized I wanted to go back to grad school. And so I ended up going back to grad school, focused on conservation, using genetic tools. But I also got at that point interested in bats and some of the emerging infectious disease work along with my PhD. So I worked on a virus called Nipah virus for my PhD, which is an emerging paramyxovirus, which is harbored by these fruit bats. I'm not going to talk so much about that specific work. I'm going to talk a little more general, I think, about things that hopefully would be of interest to you and some of the microbial work you all do, thinking about using modeling and analytics to forecast pathogen diversity, and how can we use modeling tools to sort of target our surveillance for emerging infectious diseases. But in my sort of time as like a postdoc and after, also did a little bit of collaboration on the Hawaiian hoary bat. So we did some phylogeography studies to look at where did bats come from, when did they arrive in Hawaii, how many migration events brought them to Hawaii. And this cheesy title kind of tells you a little bit about some of those findings. That there were two major, at least, migration events that established the Hawaiian hoary bats in the islands. And they came from California, basically. Yeah. So they flew a long way. They got blown off course and they landed here twice and established. So it's kind of cool story. But the story today is really about emerging infectious diseases. So as Matt nicely said, I spent 15 years working with an NGO, nonprofit, originally called Wildlife Trust, which was then later rebranded to EcoHealth Alliance. Based in New York, but all the work is international around the world, working with about 20, 30 different countries, mostly lower middle income countries, to strengthen their health systems, really from a wildlife, environmental, and sort of human health triangle, what we know of now more commonly as One Health. So strengthening those systems with the Ministry of Environment. So every government, every country we worked in, we worked with the Ministry of Environment or Wildlife, Department of Wildlife, Ministry of Health, and usually the Ministry of Agriculture, and sort of triangulated the work across that livestock, wildlife, human interface primarily. So the goal of the organization was, and I say was because EcoHealth Alliance is not really functioning at the moment. And so the timely departure brought me here, which is great news for me, but bad news for this organization. But we really did great work building these sort of science-based solutions for preventing pandemics while simultaneously protecting ecosystems and conservation. So it's a pretty unique mission, actually. There's other organizations that are working in this space, but we were some of the, I think, the pioneers working at that interface. And so one of the grand challenges we were working to solve, of course, is a very big one, which is zoonotic disease emergence. Zoonoses, as you know, are diseases that jump from animals to people or from people to animals, vice versa, can be bidirectional. And this is a map we put together early on, published in Nature, Jones et al. And I can't remember what year, but early 2000s. And it was essentially just trying to figure out, and this is old school, going through the literature, through thousands and thousands of papers in the literature, trying to figure out where was the first emergence, the first human case of a new disease. So there are about 400 emerging infectious diseases that were in this database. And what we did was look for the first known human case of each of those emergent. So HIV would be one data point on this map, right? So where did HIV first likely jump into people? And that would be, somewhere here in Central Africa, one of those dots. So that's kind of what became a baseline. And from that, you could extract other metadata, which shows clearly that emerging zoonoses are on the rise, emerging infectious diseases are on the rise, and a large proportion of them, a number we commonly cite, something like 70%, are zoonotic. So why did those zoonotic diseases emerge? Again, this is a literature review study we did, but if you look basically at what you can glean from the studies, land use change is a very big driver of new emerging disease events, as is larger agricultural industry changes. So like thinking of, high input, mass production of livestock and crops, monoculture, big changes, international travel and trade, of course, other things like war and famine and other sort of socioeconomic drivers of disease emergence. But if you take this land use change piece, it's quite a big one. It's about a third of all the new EIDs from this literature review. And that includes things like structurally changing habitat through deforestation or fragmentation, and changing the sort of natural species dynamics that happen in an ecosystem when those things are perturbed. And there's a whole field of literature on land use change and emerging disease. And we've contributed to that, but there's a lot of people that have done great work on that, including, probably some of the work on Lyme disease, right, and dilution effect and all those studies. The other big challenge, of course, is emerging diseases are not only increasing in frequency, but increasing in economic damages and costs. And we saw this from SARS-CoV-2. We're talking about trillions, if not many, many billions of dollars in damages. And so there's a strong economic argument as to why this work is important as well. And so we expanded on some of the emerging disease hotspot mapping. I'm not going to go into all the details of this, but you can read the paper if you're interested. But essentially, we took that map of all those initial emergence events, and we did a spatial analysis looking at, well, what are the correlates? What are the ecological correlates that relate to ecosystem change, that relate to human population change, all those drivers, right? Those emerging infectious disease drivers. And you can map those out spatially. And this is a map that shows you emerging infectious disease risk around the world, where it's yellow, there's a greater risk of a new emerging disease jumping from animals into the human population. So this is just for zoonoses. So, of course, this is way pre-COVID. We know it wasn't a surprise. We know that southern China, India, certain parts of Central Africa, et cetera, are hotspots for emerging infectious diseases. So this work was some of the first that sort of to try and map that out globally and look at, well, what are the underlying factors that lead to that emergence? Okay, why do we want to use a One Health approach? And what is a One Health approach? Well, the high-level expert panel, OLEP, of the United Nations, these what people call the quadripartite, which is the Food and Ag Organization, the World Organization for Animal Health, formerly OIE, the UN Environment Program, UNEP, and WHO, have a high-level panel of people that sit on it, One Health experts, and they recently, several years ago, defined One Health, kind of gave it a formal definition. And you can read that online, but this is the graphic that's included in their paper. So One Health is if you think of it, this flow, it's kind of the outcome, right? Which is a healthier planet, healthier ecosystems, healthier animals, and healthier humans, right? And those three things kind of working together. But how you get there, right, is the important part. And they call this the 4 Cs. So it's through collaboration, right, where you bring sectors together, sectors that maybe didn't historically work together very well. You're speaking the same language, you're communicating, you're coordinating so there's a lot of work on societal coordination. Think of like the Department of Transportation, right? How important it is for Hawaii for biosecurity, right? To think about what's coming in and out. So you need to coordinate at a government and state and national and global level. So there's a lot of work on coordination, inter-ministry coordination, international coordination, in order to get there. And then training, right? So training and capacity building are fundamental to One Health. How do you build the next generation of One Health practitioners? Well, you need to be trained cross-disciplinary. You need to have those opportunities for training. So that's what One Health is. And why does One Health work? Well, we know most diseases are zoonotic, right? So they come from animals. Many of them are vector borne, right? You have a vector intermediary. And you have this sort of, these things are always out there, right? There's millions of viruses on the planet. Likely. We haven't named them all, but there's likely millions of things out there. And there's many animals. We're focused on mammals a lot. I mean, birds are very important as well for emerging disease, like avian influenza. But mammals tend to share the most of their diseases with us because we're mammals, right? So there's thousands of mammals. And there's always pathogens circulating in those natural ecosystems, as you know. The opportunity for action is really here, right? When we have first initial spillover, and then the other opportunity is early detection. So maybe you catch something that looks really weird, like camels are dying and nobody knows why they're dying. Well, let's look at those camels and let's see what is in them. Are people coming into contact with those animals? What is happening at that interface, right? So that's kind of the early stage for intervention. You can also catch things early. So there's someone that's sick and they're bleeding out of their nose and they're sneezing. That looks like a weird clinical sign. How do you have astute clinicians who are trained to recognize that and then have the tools for pathogen discovery to find those new things? And then have the governmental system to say, okay, well, we need to isolate, we need to quarantine, we need to take this seriously in order to stop that human-to-human transmission. So as you go further along the sort of pandemic emergence spectrum, right, from natural animal populations to sort of spillover between animals or between vectors to human infection to human-to-human infection to global spread, it gets more expensive and it gets more impossible to control as you go on that path. So the action for One Health is here in this area, and that's where kind of we focus our energy in terms of how do you develop better strategies to address health at that early interface. Part of the work that EcoHealth did was do a lot of policy work in terms of engaging with policymakers at a national level, at an international level, and setting some of these standards. So again, this is all pre-COVID stuff, but there was the intergovernmental, I always forget this one, Intergovernmental Platform on Biodiversity Ecosystem Services, IPBES, right? That's the equivalent, the ecologist equivalent of the Intergovernmental Panel on Climate Change, right? Looking at how is ecosystem change impacting disease emergence. So there's been some big high-level global studies on this. And one of the outcomes is, again, that economic argument that if you invest billions, even if it's billions and billions of dollars into preventing pandemics at their source, you're going to save at least 2 orders of magnitude money in the damages that will be produced from those pandemics. So you look at a pandemic costing tens of billions of dollars or $1 trillion, right, like COVID. If we started investing billions of dollars more strategically into, hey, say curbing deforestation at high-risk interfaces, then we're basically putting money down to protect our health and the global economy downstream. Okay, well, how do we get there? And what are some of the other key kind of questions and open-ended questions? So one of them was really how many viruses are out there in wildlife that could potentially affect people? So this was sort of a big open question. We started to chip away at it again, something you could probably do now in like a matter of weeks, not years, with the tools of AI. But we went through with like lots of interns and lots of staff, lots of literature to pull this database together, which is essentially a pretty simple database if you think about it. It's basically every known virus that infected mammals. And there, at the time we started this, there weren't that many named viruses that affect mammals that we knew about. There were like 600 in the 2010. Yes, we started this in 2010. Taxonomy of viruses at the time that were mammalian infecting viruses. And of those, they infected about 800 species of mammals. After we went through all the literature and we pulled all the mammal species, got all the sort of validated data, that's kind of the extent of the database. Now, there's lots of interactions, right? So one virus can infect multiple mammal species. And so if you look at the raw data, and this is little silhouettes showing you the mammalian orders. The gray are wild animals, the red are domestic animals. This is just the raw total viral richness. So the biggest is like 80, and that I think those are horses, right? So horses were known to harbor 80 unique named viruses at that time. But if you look across orders and you look at these box plots, not really any huge differences in terms of just viral richness, right? The diversity. But if you look at the proportion of those viruses in each species that are known to be zoonotic or that are capable of infecting people, this is again just the raw data, but a different picture emerges where you have, well, some orders there's just very little data and all of them were just known human viruses because there's a lot of biases in these things you have to correct for, right? One of them is that, well, we have surveillance tools for viruses that infect people because we care about them. We don't have surveillance tools for, bat coronavirus 123 that wasn't really even known, but we might have a measles assay, right? So you might start testing for measles and you find, oh, some of these animals are reacting to measles. So that's some of the biases inherent in that. But nonetheless, you see bats, primates, and rodents, even with the raw data, come out as a relatively high proportion of their viruses are known to be zoonotic. And then the ungulates down here, even though they have really high viral richness on the sort of domestic animals, they have a relatively low proportion of zoonotic viruses. So they have some unique horse viruses, some unique cow viruses, but not the ones that infect us. So again, just the raw data. And there was some follow-up work published in Science like a year later that extrapolated based on this data we put together and suggested that there's, upwards of 1 million or so as of yet discovered zoonotic viruses. Okay, but the real fun part was after we put this database together, we could do some analysis. And the big question here is, well, are there certain traits that some wild animal species have, wild mammals, that make them better at hosting viruses, better viral reservoirs, or make them better at hosting zoonotic viruses or being able to transmit those viruses to us? So this is a generalized additive model. Again, I'm not going to walk you through all the details, but essentially these are like partial effect plots. So it shows you the relative influence of each variable. And these are all the significant variables that were included in these panels. So the top row is for viral richness. So after you control for research effort or the number of disease-related publications per species, you see that body mass matters in a sort of nonlinear function. The size, the range size of the species. So those with bigger ranges host more viruses, probably because they're exposed to different environments and they're exposed to other species. And the number of other mammals that overlap with them seems to matter. And then irregardless of that, you have certain orders of mammals that have more viruses than other orders after you control for all these other things. And the bottom row is the proportion of zoonotic. So the proportion of viruses that are known to infect people. And you have two things that are important after you control for publications again. You have this phylogenetic relatedness to people. So there's a weird kind of curve there, but basically the more closely related you are to a human, the more viruses you share. So chimpanzees, we share a lot of viruses with them. And the more of your range, your natural home range that overlaps with human density and human populations, the more viruses you share with people. So there's kind of this effect of relatedness and of contact, right, at a very large global scale. And then again, this is a result that got a lot of interest, and it's a very, not just above the significance line there, but bats, despite all the other factors, when you control for everything else, seem to have a higher proportion of zoonotic viruses than the other orders of mammals that were included in the analysis. And we can dive into that in a second. So then you can take the outputs of those GAMs and you could basically forecast what should be the number of viruses in the Hawaiian hoary bat. You know, and my model, based on body weight, based on relatedness to people, based on et cetera, based on like the research effort to date, we expect there's like five viruses in the Hawaiian hoary bat. But actually, we look in the literature and there's 0 that have been discovered. So what we called those in the paper is the missing zoonoses. So those are the ones that we forecast, right? The model predicts that the species should have X number, say, of zoonotic viruses, but we don't know of any, or we know of 1. And so you take the difference and there's 4 that we haven't found yet, right? So those are the missing zoonoses. So then you can map those out based on each species range. And you can look, where on the planet are there a rather large or an aggregate number of as of yet undiscovered viruses or as of yet undiscovered zoonoses. So these are our missing virus or missing zoonoses maps, and you can look up at them by each order. And you can dive deeper into the data too. This is like, I don't think this is even in the paper actually. But this is like just for the bat finding, the missing zoonoses by bats. It varies dramatically based on what family of bat you're looking at, right? So different families also have different factors. So within an order, you have variation as well, within an order of mammals. And then there's also a lot of important information in the missing information here. So like I said, we only had 800 species of mammals in our database because only at the time 800 species of mammals had had at least 1 virus known from them. But there were hundreds and hundreds, thousands of animals where no one had ever found a virus, no one had ever done surveillance, right? So you can use these maps as well. Or you want to go find new bat viruses that have never been discovered, go to Latin America, because there's really high bat richness there and hardly anyone's looked, right? So we forecast there's a lot of things that need to be found. So we built off of this work and looked at sort of, this is with Greg Albery, 2020. Not only thinking about people as the end question, right? The other work was kind of focused on zoonotic viruses infecting people, right? It's a very human-centric view. Probably why I got into nature. This one got into nature communications, but it was looking at sharing across all mammals, which is almost a bigger question, right? How are viruses shared between bat species A and rodent species B, right? And why are they shared? And again, it's that those same 2 factors that came out really were a very simple model, but actually explained most of the variation, was phylogenetic relatedness. So if those 2 mammal species are closely related to each other genetically, they're going to share more pathogens. And if they overlap, they're going to share more pathogens. But these are like nonlinear relationships. So you can look at, okay, for a given geographic overlap, what do we expect the effect of relatedness to do, right? That's what this plot is showing. For a given relatedness, what do we expect a certain amount of geographic overlap to look like, right? And you can begin to develop some predictability in terms of viral sharing. This work was then extended into another study with Colin Carlson that looked at, well, what will happen with climate change in the future? So if we map out all the mammal species that occur globally, and we account for future projections in climate change and land use change, it included land use change as well. So what's going to happen, right? Well, we're clearing land, places are getting hotter, mammal ranges are changing. Some species can move more easily than others, right? So for each species, they're going to have a different ability to migrate and move. And so you can look at those changing patterns of distribution and combining it with that phylogeographic model that I just showed you, you can say, well, when those new species overlap, what's the chance they're going to share new pathogens? Right. So this is kind of like a look into future viral sharing with climate change across all mammals. And there's going to be certain hotspots where viruses are shared that look different than, say, the emerging human infectious disease maps, right? But these are areas where with climate change increasing, you're going to see a lot more species coming together. Some of the highlands of Indonesia, where there's a lot of mammal diversity to begin with, right? You're going to see new species coming together and sharing diseases for the first time. We are going to be the unfortunate beneficiary of those sharing events as well, right? Because as more species start sharing, then they develop evolutionary propensity to start jumping into new hosts, etc. So this is an important first step, but there's more work to build on that. I'm sure a lot of you are genetic people, so we've also used phylogenetic approaches. So based on all the surveillance data we've acquired, where you go out to Indonesia and you sample a bunch of bats and rats, and you see, oh, what new viruses did we find? And then you sequence those viruses and you identify the host. Well, then you can start mapping out things like this, which is just a phylogeographic network. And in this case, it's important data because it's all the SARS-related coronaviruses that we knew about at the time in 2020. So this is Rhinolophus horseshoe-nosed bats, are all these R species. These are Hippocyterus bats, which are related. So the pie is color-coded by the host, but the network is showing you the virus network and all the different, bottom line, there's a huge diversity of SARS-related viruses. This is the clade that contains SARS-CoV-2, and this is the clade that contains SARS-CoV-1. Remember SARS-1? Yeah, we dodged a bullet on that one, but COVID we did not. But yeah, about 800 people died of SARS-1, something like that. So there's a huge diversity of natural viruses circulating out there. So we can use this, and I don't have the next slide that I normally would have here, but we can use this to say, okay, well, what hosts are most important, right? If we care about SARS-CoV-2 related viruses, are there certain clades of mammal hosts that only carry those viruses? We can also look at what is the propensity of those viruses to jump between different hosts, right? To host shift based on the genetics. And then, we did a follow-up study after COVID first emerged, which was trying to estimate, okay, well, we know that all these other SARS coronaviruses are out there, and we have been doing surveillance on this since SARS-1, actually, right? So when SARS-2 emerged, the question is, okay, well, what's the chance SARS-3 and SARS-4 are going to emerge, and where is that going to happen, and what species is that going to come from? And so this is an exercise, a probabilistic risk model exercise, where we looked at the strategy, or we looked at a strategy, I'm not saying this is the final answer, to assess spillover risk of future SARS-CoV viruses, SARS-related coronaviruses. And the bottom line is, when you put all the risk factors together, so this is the range, this is the SARS coronavirus related the distribution of hosts that carry SARS-related coronaviruses. So where it's redder, there's like a stack. There's like 14 bat species that carry SARS-related viruses, right? So that alone is not risk, but it's telling you, well, there's a diversity of hosts and a diversity of viruses in that area. And then you overlay human population density on top of that. And then you overlay information you can gather of of bat density, if you can get that, right? Are people coming into contact with bats? And then you can overlay human behavioral data, questionnaires. We go out and we ask people, do you eat bat? Do you hunt bats? Do you see bats roosting around your house or your workplace? And you can incorporate those sort of questionnaire data into this probabilistic risk model as well, and say on average, in a given village in southern China, about 1% of people, see bats on a weekly basis. I'm making that number up, right? But you can incorporate that and then you add it all up. And at the end of this exercise, adding up those probabilities, there's a non-zero probability that we will have that people are infected by SARS coronaviruses every year. It might be as many as 66,000, which sounds crazy. It's a lot, but the confidence intervals on this are huge, right? So it's something that's non-zero, which means these viruses are spilling over on a regular basis. So the empirical data have supported this, and as people are going out there and they're doing serological surveys in the human population, we see that, oh, this population that was not even close to where SARS-1 emerged, they have antibodies to SARS-related viruses, right? So we looked at archived serum from before COVID, because COVID's kind of made this very difficult. Everyone's vaccinated or exposed, right? But when you look at archived serum from before COVID, you see, oh, there's pockets. People are being exposed to these viruses in Indonesia, in Myanmar, in Thailand. There's multiple studies that have shown this. So, if anything, these modeling exercises point to where you need more data, which is why we still need field biologists. So don't worry, you're not out of a job yet, if you're a field person. So, right, we need more data of like the viral diversity that's out there, the prevalence in hosts. We work with veterinarians and wildlife biologists and get out there on the ground to collect that data. The role of potential bridge hosts or intermediate hosts and how you can quantify that. And then more sero surveys, as I was just talking about, sort of ground truth and use this as a way of looking at when is spillover actually happening. And then just some pictures, this is some from my PhD and postdoc work in Bangladesh, looking at Nipah virus and other viral surveillance projects we've done. How am I doing on time? We go till
we go to one.
till 1?
Yeah.
Oh, my watch keeps winging out. Oh, there we go. 12:36. Okay. So part of this, part of our work was supported by USAID, rest in peace, which was is an amazing institution and had amazing programs on global health that have mostly been gutted, unfortunately. But one of those big programs is called the Emerging Pandemic Threat Program. I guess I shouldn't get too political, but yeah, very important program, trying to prevent emerging pandemics around the world, right? And strengthening global systems to do that work in the country, which ultimately is protecting the homeland of the United States of America, because diseases that emerge anywhere pose a threat to the United States, right? So it was really global security, which is also national security, to build some of this capacity for zoonotic disease preparedness. So I worked on this project for 10 years. We worked in 30 countries. We trained over 6,000 One Health professionals. We strengthened capacity in 60 labs to do the sort of viral diagnostic work. We sampled 160,000 animals and people, and we found about 1,000 viruses. And a lot of these were small genetic fragments of viruses, not full genomes, but there were quite a lot of new discoveries as part of that work as well. So Predict really operationalized a lot of the modeling I was discussing. So in fact, so a lot of this modeling was developed as part of the Predict modeling and analytic team that I helped run, so that we could collect data from our field operations and feed into the models, but also develop the models to help make some decisions of where should we go do the field work, right? So like the idea is iterative, right? The models sort of defined, well, what species should we sample and where should we go? And then as we collected data from the field, those were helping to refine the models in terms of thinking about risk and new diseases, viral diversity, risk, human exposure, et cetera. So for those ecologists in the room, I think if you've done species discovery curves, right, as part of at least your studies. So we applied this. This is not the original paper, but I think I don't remember what year it is. Simon Anthony et al., 2012 or 10 or something. We had a paper that looked at basically using it for the same thing for viral discovery. And it was the first time anyone had actually used that approach for viral discovery. But if you start repeatedly sampling the same populations or the same species over time, you can get an estimate of what is the viral diversity in that population, right? Just like you go into a forest and you go mistnet for birds over and over again, and finally you're catching all the birds, well, you know that your population is 100 birds, right? Same idea. So we did the same thing, these kind of like mark recapture studies to get an estimate of viral diversity. And you see, for some families of bats, you have like coronavirus diversity, like 20 species estimated. And many of these, aren't even tapering off, right? So diversity, it gets rarer to find the rarer viruses, but you can kind of get an estimate of the global diversity as well. Another paper kind of use if those of you have insurance, you have like your insurance score. That was the idea here, right? You have like a little meter where you look at your risk. Should I give this person flood insurance if they live on Kailua Beach? Maybe not. So we looked at kind of a suite of factors. So the overlying question is, okay, we discover 1,000 new viruses. How do we know which of those we need to prioritize? Well, you can look at other information about related viruses, right? Like nearest relative type work. So there's viral traits, right? There's viral traits that might be predictive of whether it can be a human infectious virus or a pathogenic virus. So, and now with AI and genomics, that's changing, right? There's new tools for this. So you can run the genomes through AI. You can use other approaches people have been using to look at risk based on genetic markers. But nonetheless, this is kind of like crude traits. You can look at where the virus came from. What species did it come from? Do people interact regularly with that species? Is there a risk there? Does the species overlap with a lot of human villages and human population? You can look at the ecological context. Is it found in a deforested area or a primary forest? So the idea was to take all these host, environment, and virus traits and kind of create a quick, rough estimate of whether or not it's zoonotic or whether or not it could have impact. So again, this is like a first stab. It's something that there's new tools now to do this stuff better. But I think it's an interesting idea. And then, there's time and place for very targeted disease surveillance. I'll just give you a taste of some of the work I've done before. So when MERS, Middle Eastern Respiratory Syndrome coronavirus first emerged in 2012, we went out to Saudi Arabia to try and figure out where this thing came from. That was really interesting work. I could tell you lots of stories about it. But essentially, we found some evidence that there was one bat species that had MERS-related virus in it. But it was very preliminary. It was one animal, actually, and it got a lot of press because people were hungry for the answer. But the real answer came out later, and that was our team's work as well, was part of that, is that camels were really the way people were getting infected. Right, so MERS, based on the evolutionary history and other information, looks like it was a bat origin virus, jumped into camels, probably quite a long time ago. Well, decades ago, not like in this time frame. And maybe it happened in East Africa, and then through the camel trade and through human exposure to camels is where people get infected from MERS. Luckily, MERS does not seem to be human-to-human transmissible very easily. So that's a saving grace, right? But this is the kind of thing that you kind of have to do this detective work, right? To trace back where something came from and what is the real risk of it in the future. So MERS led me to an interest in and a realization that there's a huge gap in the Middle East in terms of disease surveillance at the wildlife interface. And so we started this program with DoD funding called WabiNet, which is a Western Asia Bat Research Network, focused on understanding the risk of new bat emerging diseases in the Middle East in particular. So it's kind of a dual goal. One was to collect actual surveillance data of what's out there. So are there other MERS-related viruses out there? Can we find MERS in some of these bat populations? And then secondly, really to build capacity to strengthen relations, collaboration across the countries. Really a great project. It was a great learning experience for me. It lasted 5 years. Unfortunately, we didn't get a renewal, but that ended a couple of years ago. And then there's just some pictures from the field, from some of the countries we worked in. And then we did these sort of field to lab training opportunities where we did cross training. So here we have our Omanis in Jordan, and here we have our Turkish and Jordan colleagues in Georgia, the country of Georgia. And then we did collect quite a bit of data, and we also use that to sort of refine some of the risk maps. Are there bat human emerging risks in the Middle East? And where does that risk vary? I mean, there's a lot more diversity in Turkey from what we found, for example, and you can kind of look at that. These are all, so there are about 20 coronavirus bat species that we sampled. Okay, a really critical part of all this, obviously, and this is true everywhere, is engaging with the local communities, right? The people that are selling rats in the markets in Indonesia, they're sort of the front lines, right? They might be the ones who are exposed first to a new rat virus, right? Well, they might not be the first. The hunters would be the first, but they might be the second in line. And so we've worked a lot with the local and indigenous communities in the countries we worked in. This is a study we published in Indonesia that shows you the trade routes. So there's a lot of wildlife hunting going on, and this is kind of just showing you where the animals have moved over time. And then we did the sort of behavioral aspect, right? So looking at, well, how are people interacting with these animals? So that heat plot is just showing you the result of a bunch of surveys, right? We went around and we did surveys with people, and this is showing you, it's hard to see, there's some faint boxes there, but like rodent interaction in the house, okay, that's known. But if you look at bats, there's actually very faint, but there's actually a fair number of people that interact with bats at slaughter, because we're working in some of these markets where they eat bats and they eat rodents, primates. And then you can collect really important qualitative data too, right? Which gives you an understanding of the sort of knowledge, attitudes, and practices, right? So do people have a general understanding of emerging diseases? If I'm butchering animals and I have an open wound, is that a problem? Many people said no, right? But so that gives you a baseline of where to start if you're going to bring interventions, say, into the market. Say an intervention is a hand washing station, right? Will people use the hand washing station? Will that prevent pathogen exposure? That kind of thing. And then we developed some community outreach tools. So this was a book called Living Safely with Bats, which was really meant to get a dual message out there. One, bats are good for the environment. Don't go out there and kill bats. Two, if bats are living in your house or your community, here's ways where you can live safely with them to reduce your contact, to reduce your exposure. So we worked with behavioral scientists and community members to sort of develop, translate, and vet these tools out in different places. Here's a version of Living Safe with Wildlife. We translated into Visayan for a Philippine project that I was leading. I'm going to skip over this. Okay, so that's the end of that. I can answer questions. That's a summary of some of the work that I had done at EcoHealth, kind of the big picture of how do we predict zoonotic viruses, how do we discover them, and how do we mitigate their introduction into people or other animals. But just kind of in my current role back here at home, wanted to just give a little bit about, where I think the work you guys are doing, and I've read a bit, through the C-MΑIKI website and some of the great work you guys have done. Hawaii is, I think, a perfect model for really integrated One Health. And there's a number of reasons for that, but I think it's probably obvious to you and hopefully people here at UH, right? I just stole that off the web from the UH One Health certificate page, right? So I think there's a growing interest in One Health among the student population, among the professors that I know that I'm engaging with, right? There's a growing interest in applying One Health approaches to Hawaii. And I think that's great. And I want to see more of that. So I'm trying to steer CTAR into doing more One Health work as well. I think it's natural to think of this sort of Ahupua'a based system as like a perfect integrated One Health system where you have natural ecosystems up in the mountains, right, with very little human impact. You have intermediary areas, include agricultural production systems, and then you have more urbanized areas, local communities, right? And if you think about how all these are connected, and then on top of it, you have the opportunity in Hawaii, which other states don't have, which is really linking terrestrial, marine, One Health together. And, there's a number of pathogens that cross that barrier, which I think are prime for this. And there's, again, the traditional knowledge of the Ahupua'a is that all these things rely on each other, right? And the same is true when there's dysfunction or disease, those effects trickle down from mountain to sea. And of course, the Ahupua'a doesn't look like that anymore, but it looks more like this, right? And so in our urbanized and our more modern environment, even here on Oahu, we still have these opportunities to explore those systems. And I think that's really an opportunity. And I think not only within CTAR, but across all the colleges, we can collaborate and do a lot more from that One Health paradigm. So for those of you that aren't familiar with CTAR, I encourage you to learn more about us, but we're quite a diverse college. And I think you've probably gathered that if you know some CTAR folks that are in different departments. So CTAR includes fashion design and merchandising, right? So family consumer science department includes our fashion department, includes our family science department. We have, and these are just some of the majors that are initialed here, but we have the Department of Molecular Biology and Bioengineering, right? Which is, I guess, the test tube there. We have Natural Resource Environmental Management, NREM. We have Tropical Plant Soil Science. We have Plant Environmental Protection Science, PEPS. We have Human Food Nutrition and Animal Science. Right, all these historically were more than 6 departments, but they even merged those. So many of these departments are actually merged from 2 other departments. So it's really this huge diversity of work. So one of the things I've been thinking about, and this is actually a slide from my job talk, is how do we really pull this huge diversity of work that's happening at CTAR together in this sort of approach where we're strengthening communities, we're strengthening agriculture, and we're strengthening natural ecosystems. And again, I think the One Health paradigm is a cross-cutting theme that really brings those together. And so there's huge opportunity for us internally at CTAR to work together through that lens, and certainly collaborating with other colleges and other campuses at UH as well. And I think plenty of you know that CTAR recently changed its name about 1.5 years ago from College of Tropical Agriculture and Human Resources to Human Resilience. And I think this really fits in with building that human resilience as well. And then we have this huge opportunity in these 22 plus research stations around the state that CTAR manages, which are our applied agricultural research stations and extension stations. And so we're really working now to bring new life into these stations, make them available for student training, for more faculty engagement, improving the infrastructure on the stations. There's been a lot of investment in the last few years on that. And so, encourage you to collaborate with us on, opportunities at our research stations as well. I think it doesn't have to be just within our college. I think there's opportunities more broadly that we can leverage. And then lastly, I guess I'll just finish with another project I'm working on, which is digitizing. So CTAR is the founding college of the University of Hawaii. 1907, we started in 1907. And since that time, we've been generating knowledge, much of it on paper, not digitized yet. So one of the big things I'm pushing for and working on now is really digitizing this extensive library of extension publications and research publications so that we can create a digital accessible library and database, which of course can be powered through AI that you could query, they could pull data out of, and ask questions, how have pest management strategies for sweet potato changed over the last 50 years? And you can quickly query that huge library of knowledge and generate some outputs. So this is in process. We're working on hiring a grad student now to work with the University Library Archives to start archiving the CTAR library better. And then we'll start on the digitizing process to pull that in. And I think, yeah, mahalo. I hope there's time for a few minutes for questions. Thank you.